DNA Replication by Jacob Osterloh and Tyler Perkins. DNA double helix is the structure formed by two strands of DNA. Those gray pieces are nucleotides, which are the monomers of nucleic acid. Nucleotides are made up of nitrogenous bases, which are adenine, thymine, guanine, and cytosine in DNA. Genetic information is stored as those. Nucleotides are made up of nitrogenous base, deoxyribose, and the phosphate group forms the sugar-phosphate backbone, which is the structural framework of nucleic acids. This is held together with covalent bonds, also called phosphodiester bonds. Here, we see the helicase unwinding the DNA double helix. Helicase unwinds the DNA double helix by breaking the hydrogen bonds. The separation of the strands in a Y shape is called the replication fork, and it starts at the origin of replication. The origin of replication is a specific sequence of nucleotides where the DNA replication process begins. One strand is oriented 3' to 5', which is the leading strand. The other strand is oriented 5' to 3', and this is the lagging strand. This is an effect that we call anti-parallel, because the strands run opposite of each other. Single-stranded binding proteins now bind to the single strands to keep them single. DNA gyrase now comes in and adds NICs to the sugar-phosphate backbone. RNA primase now adds RNA primer on the parent strand. The primer acts as a starting point for DNA synthesis. DNA polymerase binds to the leading strand and walks along it, adding new complementary nucleotide bases to the strand of DNA in the 5' to 3' direction. The sliding clamp prevents the DNA polymerase from dissociating. RNase H removes the RNA primer. Now the lagging strand is replicated. RNA primase adds RNA primer. DNA polymerase now replicates the strand in 5' to 3' direction, because that's the only way DNA can be replicated. RNase H now removes the primer. The previous process repeats. These segments of DNA are called Ozaki fragments. The DNA strands are sealed and the NICs are removed by DNA ligase. After that, telomerase adds repetitive nucleotides to prevent genetic material from being cut off. These aptly named nucleotides are called telomeres.